Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's Monday Seminar Series. Before introducing today's speaker, let me remind you to ask your questions for the speaker using the Q&A icon located on the webinar toolbar. Please do not use the chat box. You can add questions at any time during the presentation as they arise. There is no need to wait until the end of the presentation. Today's speaker is Dr. Yvonne Nolan, a professor of anatomy and neuroscience at the University of Cork College. Dr. Nolan is also an APC Microbiome Ireland funded and investigator and a Science Foundation Ireland investigator. Her group examines the effects of inflammation and lifestyle factors such as exercise, stress, and diet throughout the life course and its impact on neuronal function and behavior, with a focus on the role of the gut microbiome and hippocampal neurogenesis. And today, Dr. Nolan will discuss the research in a presentation titled The Role of Gut Microbiota in Hippocampal Plasticity in Alzheimer's Disease, Lessons from Fecal Microbiota Transplantation Studies. And with that, I will turn the microphone over to Dr. Nolan.
Thank you, Paul, and thank you to Peggy and to all of you for inviting me to speak with you today and to share some of the results and some of the research that we're doing. So I'm going to focus in on the role of the gut microbiota that we've been investigating and specifically in the context of Alzheimer's. I'll also touch then on some of the work that we're doing in the context of exercise and how we believe that the gut microbiota and brain plasticity processes are potentially mediating some of the effects that we see of exercise on brain and brain aging. Okay, so just by way of context and to start, this is information that many of us and many of you will know that about our life expectancy increasing, and the predicted average life expectancy to increase could be we could live to age 110 by 2050. And we also know from statistics from the World Health Organization that there are now more people in the world aged over 65 than aged under 5. And this is due to the increases in life expectancy, but also due to falling fertility rates. So a consequence then, when we think of brain aging, of this aging society and these numbers and statistics that we have, is the risk for development of neurodegenerative disorders is massively increased. So aging is the biggest risk factor for the development of dementia. And so dementia is an umbrella term we used for an impaired ability to remember, and this then interferes with our everyday activities, and it's common as we age. And Alzheimer's then is the most common type of dementia. So here are some, again, statistics around dementia from the World Health Organization. And so as our societies age, the number of people living with dementia then is expected to rise. So current estimates are about 55 million people with dementia in 2019, that by 2050, this will have almost tripled to 139 million people globally. So while our life expectancy is longer, we're living longer, not everyone, as you're well aware, is aging healthily. And then this increases also the risk for developing dementia and Alzheimer's. It also puts pressure on our healthcare systems, and so it has economic as well as health implications. And again, more figures, more facts. The global costs now associated with dementia itself is also set to double. So an estimated of 1.3 trillion US dollars in 2019 to go to 2.8 trillion in 2030. So really what this is setting the scene for us is that we really need to understand and implement factors to try and prolong healthy aging. And related with this then is to delay or reduce the risk of developing dementia and Alzheimer's disease. So what I just want to show you here is, we know that determinants of healthy aging include genetic, environmental, lifestyle factors, and research over decades now has identified a number of modifiable risk factors for dementia in particular. And these include things like diabetes, obesity, depression, physical activity, and especially diet. And this means that by acting on these, then it may be possible to delay or reduce the risk of developing dementia. So from this report from the Lancet a couple of years ago, it's been proposed that up to 40% of dementia cases could be prevented or delayed by addressing these 12 risk factors. And so what's interesting for us and for the work we do and the research that we do is that many of these risk factors are associated with changes in the gut microbiota. So especially factors like diet, like exercise, like disease course, and of course, aging itself. So by determining associations and by researching the causal roles between gut microbiota, between these lifestyle factors, aging and disease, it's really becoming an area of interest, of scientific interest and research. So a brain structure then that's particularly vulnerable or receptive to these modifiable lifestyle factors is the hippocampus region of the brain. The hippocampus is responsible for learning, for memory, but also for regulation of emotion. It's also one of the first and one of the primary brain regions to be affected in Alzheimer's. So clinically then Alzheimer's is characterized as having deficits in memory, in cognitive abilities. This can lead to a decline in mood as well as functional activities that impact quite severely on quality of life. And progressive in nature. So there's progressive neuronal loss, progressive loss of synaptic function throughout the brain, but especially in the hippocampus. It's also characterized pathologically by accumulation of plaques and tangles. And so these are the classic pathological or hallmark features of the brain, which can be identified post-mortem. But Alzheimer's is really considered to be more than that, and it's considered as a multifactorial disease now. And this is because of the interactions that we know and that research is informing us between genetic predisposition, but also the interactions between lifestyle factors and environmental factors that can influence the disease course. So a number of peripheral mediators now, including inflammatory molecules and gut bacteria-derived products, such as metabolites, are influenced by environment, by lifestyle, and are increasingly becoming recognized as mediating changes in brain and behavior, and especially on cognition. So these gut microbiota then are these trillions of microorganisms that inhabit the gastrointestinal tract, are becoming a target for investigation for brain aging and for diseases of brain aging, such as Alzheimer's disease. So like the hippocampus, the gut microbiota are very responsive to lifestyle and environmental factors, such as diet, exercise, also stress and inflammation negatively. And so we think then that the gut microbiota could be a peripheral route through which these factors could impact upon changes on hippocampal function, on behavior, and could potentially contribute to the development or prevention or delay of Alzheimer's. So it's been shown from a number of studies that Alzheimer's patients display alterations in their gut microbiota composition. So a recent systematic review and meta-analysis, I think there were 34 cases of gut microbial characteristics in Alzheimer's, has been published and shown that the relative abundance of a bacterial phylum Firmicutes, which produces beneficial bacteria, bacterial metabolites, that this was lower, the abundance was lower in Alzheimer's patients, whereas there was a higher abundance of bacteroidetes, which are inflammatory in activity. And this has also been shown in cases of MCI as well. So our collaborator in Milan has also shown that Alzheimer's patients have an increased prevalence of pro-inflammatory genera in gut microbiota, and that this positively correlated with pro-inflammatory cytokine expression in the blood. So the evidence then from human populations. These shifts and changes or dysbiosis have also been shown in mouse models of Alzheimer's. And these mouse studies and preclinical studies have shown that an altered microbial composition may contribute to the symptoms of Alzheimer's. So, for example, gut microbial transplantation from the 5XFAD mouse model of Alzheimer's into wild-type mice has been shown to increase inflammatory markers in the gut, as well as in the brain and the blood, as well as inducing memory impairments. Similarly, transplantation in the opposite direction, so this time, say, from wild-type mice into the APP-PS1 mouse model of Alzheimer's, has been shown to reduce beta amyloid deposition in these mouse, as well as to induce impairments in spatial memory. So these are in mouse models, but we were interested then in whether transfer of gut microbiota from Alzheimer's patients into rodent models could bring about some of these changes. So we asked the question then, is there a causal role for human gut microbiota in behavioral and cellular changes associated with Alzheimer's? And to do this methodologically, our question then was, does fecal microbial transplant, or FMT, from Alzheimer's patients to healthy young rats impact cognitive function? So really a proof of principle study is what we set out to determine here and answer these questions. So we carried out a study with our collaborators in Milan, and Maria, as I mentioned, and also in King's College in London. And this was under a network of centres of excellence in neurodegeneration, or COEN initiative. And the results of this we've just published last month in Brain. So Anna Maria, her group and clinical colleagues in Milan, they recruited 64 Alzheimer's patients, and this was based on clinical profile and cognitive function using the Mentimental State Exam, or MMSE, and also clinical dementia rating. And then they also had 69 cognitively healthy aged match controls in this study. And the first thing they did was to characterize the peripheral inflammatory status and also the gut microbial composition in these participants. So we saw that the plasma levels of pro-inflammatory cytokine IL-1 beta was increased. Also, the inflammasome marker NLRP3 and macrophage migration inhibitory factor, or MIF, was increased in the Alzheimer's subjects. And this was coupled also with an increase in fecal calprotectin, inflammatory marker in stool, so indicating increased systemic and gut inflammation in the Alzheimer's cohort, and this supported Anna Maria's previous findings. So looking then to the gut microbial composition, we did 16S ribosomal RNA gene sequencing and showed that the phylum levels of Bacteroidata was increased. There's a higher abundance. So again, indicative of inflammatory activity or pathobiont activity, and a lower abundance of the Firmicutes. And as I mentioned previously, this is in keeping with the previous results that have been published. At genus level then, there was a reduction in the abundance of Coprococcus. This is a genera that produces short-chain fatty acids, and in particular butyrate. And this has been shown to be associated with healthy aging in general. We also saw an increase in desulfovibrio, which is a pathobiont or potentially harmful microbe. So when we looked then at the associations with the clinical status of our participants, we saw that there was a correlation between these readouts and the MMSE score, or the the clinical score of the Alzheimer's, cognitive score, excuse me. So positive correlation was determined between this health-associated Copracoccus and the cognitive MMSE score, and a negative correlation then between the pathobiont, desulfovibrio, and the MMSE score. So we wanted to see then the potential functional contribution of the human Alzheimer's gut microbiota to any potential behavioral or cellular changes. And so Dr. Stephanie Gabruker, she was a postdoc in my group, she led this work, and she transferred gut microbiota via fecal microbial transplantation, or FMT, from a subset of these Alzheimer's patients and from the age-matched controls into healthy young rats. So Stephanie transplanted fecal samples from each donor into 4 recipient rats. Thus, we had an N of 16 then in each group, and these were wild-type Sprague-Dawley rats, and they had undergone a gut microbiota depletion protocol with antibiotics. So she confirmed then that there was an average of about 40% of the taxa from the human donors engrafted into the recipient rats. And this was determined when the microbiota were assessed about 10 days after the initial inoculation, and again at day 59, which was the end of the study. So really demonstrating that there was a shift in the gut microbiota composition towards the human material that was transferred into the rats. So then we assessed the composition, the diversity of these gut microbiota, again at that 10-day time point after the FMT and at the end of the study. So what we saw that there was a decrease in the diversity in both our cohorts, the Alzheimer's and the control group, and at both time points indicated here. And this shows a loss of bacterial species, which wouldn't be unusual after FMT. And looking then in more detail at the genera affected after the FMT, there were greater differences in the Alzheimer's colonized rats compared to those receiving the control FMT. And again, noting that there was the abundance of the desulfovibria, this pathobiont, was changed and reflected what we saw in the stool of the Alzheimer's patients. So the next thing then that Stephanie did, she analysed the morphological features of the gut in the rats, and she saw that the colon length had decreased, that the crypt depth of the gut epithelium in the colon was increased, and that the number of goblet cells was decreased in these crypts. And the goblet cells, then this result suggests that there's an altered mucin production in the gut after the FMT. And so this then may reduce the physiological or protective function of the gut to any potential invading pathogens. Stephanie then carried out a series of behavioural tests in these recipient rats, and this started 2 weeks after the initial human donor colonisation. And so what we wanted to do was to assess functions associated with the core symptoms of memory impairments in Alzheimer's disease, but also comorbidities such as anxiety, depression, and hypolocomotion. So we saw that there was no change in locomotor activity as assessed by distance moved in the open field arena. There was no change either in anxiety-related behaviour in the elevated plus maze or in antidepressant-like behaviour in the 4-swim test, as measured by immobility, swimming and climbing scores. So results here indicating that there were no specific effects of this Alzheimer's human gut microbiota on these comorbid features of Alzheimer's in these rats. We saw that, however, that spatial memory was impaired in the Morris water maze test after the Alzheimer's FMT. So rats that received the Alzheimer's FMT showed slightly impaired spatial learning, and this reflected in this increased latency to reach the platform during the training days, And then also rats with the Alzheimer's FMT spent less time in the target quadrant during the probe trial test in the Morris water maze, and this indicates an impairment in spatial memory. Also consistent with Alzheimer's-related cognitive deficits, we found that rats colonized with the material from Alzheimer's patients performed slightly worse in a novel object recognition memory task. And we also assessed a cognitive function called pattern separation, and we used this task called a modified spontaneous location recognition task. And this was devised by Lisa Saxeda's group when she was in Cambridge a number of years ago. So it uses the ability of an animal to discriminate between familiar and novel locations after a delay period. And the premise is that animals can pattern separate if they can discriminate between a new location in this small separation paradigm. And this is reported to have a higher cognitive load, or to be a harder task, essentially. So we also carry out this test using a larger separation paradigm, again, a relatively easier task. But in both cases, we saw that rats with FMT from Alzheimer's patients had an impaired ability to distinguish between these familiar and novel locations after this delay period of 24 hours. So indicative of an impairment in pattern separation. So the small separation paradigm, as I said, is models of pattern separation, and it has been shown to be reliant on a type of neuroplasticity called adult hippocampal neurogenesis. And this is a type of cellular function that we are interested in our group. So just to bring it back then, the behavioral and the translation of behaviors to Alzheimer's and to the human population. So this ability to pattern separation is an ability to pattern separate, is an ability to distinguish between highly similar events or environments. So an ability to distinguish memories that have what we call high contextual overlap in terms of space or time. And a common example then is like, how do you find your car in a different spot in the car park every day? And this ability has been shown to be impaired in Alzheimer's, and especially in early Alzheimer's disease. And recent reports have also shown that this impairment correlates with early indicators of Alzheimer's pathology in cerebrospinal fluid, CSF, such as increased beta amyloid burden and tau levels. So as I mentioned, this ability, this cognitive ability, is reliant on this type of brain plasticity called adult hippocampal neurogenesis. Other types of behaviors shown to be reliant on this function are also spatial memory and contextual memory, and also neurogenesis has a role in the regulation of emotion through antidepressant actions and in regulating anxiety behaviors and buffering the stress response. So this process then of neurogenesis occurs in the hippocampus, and specifically in the dentate gyrus region of the hippocampus. And here reside a pool of neural stem cells. And this is in what we call the granule cell layer region of the dentate gyrus of the hippocampus. And so here these neural stem cells then, they proliferate, they migrate out through the granule cell layer, and they send dendrites out into the molecular layer region of the hippocampus, and they can receive excitatory input from the entorhinal cortex and integrate into the host circuitry of the brain. So just to point out that we use, and it's commonly used in the field, this DCX, a marker of neurogenesis, and it's often used to measure neurogenesis. So what we did in this study then is we assessed neurogenesis in the hippocampus of the rats, our groups of rats. And we do this by assessing the survival of the new neurons. So we stain for BRDU, and this is a thymidine analog which incorporates into the cell cycle, and which we had injected at the start of the study. And we co-stained then for a neuronal marker, NUN. So we saw that the numbers of these co-localized BRG-NUN positive cells was reduced in the hippocampus of rats that received the Alzheimer's FMT, indicating a reduction in the survival of these new neurons and hence neurogenesis. So we also stained for this DCX, this other marker of neurons, and it stains not just the cell body, but also the neuronal processes. And so what we can do by using DCX is to classify these new neurons into neuroblasts, differentiating neurons, and immature neurons based on the degree of morphological development. And so we saw that there was a decrease in the numbers of these DCX positive cells across a number of morphological phenotypes of these cells and in total. So looking a little bit more closely then at the morphology of these new neurons, these new neurons produced in the hippocampus, they've been shown to functionally integrate into the host circuitry and to contribute to the ability to recall memories. So a requirement for this then is the dendritic sprouting and proper arborization into the molecular layer of the dentate gyrus in the hippocampus. So Stephanie then, she measured the length and the complexity of these new neurites on the DCX cells, and using 3D reconstructions and Scholl analysis, and found a decrease in the length of the neurites of these neurons, and also by quantifying the number of the dendritic intersection as a member of the as a measure, excuse me, of the dendritic complexity, also found a decrease after the Alzheimer's FMT. And so again, indicating a potential reduced capacity of these neurons to fully functionally integrate into the hippocampal circuitry as a result of fecal microbial transplant from the Alzheimer's patients. So we also looked at microglia. These are the immune cells in the brain, and specifically we looked at these in the hippocampus by carrying out IBA1 staining. And so again, very subtle increases in the cell somal area as an index of the microglial and the immune activity. Also, we saw that there was a shift in the percentage of cells having a larger cell somal area of microglia after the FMT from the AD patients. So really there wasn't a strong phenotype of neuroinflammatory processes in the hippocampus, suggesting that perhaps neuroinflammation may not be a significant mediator, at least in the context of the work that we did here and in the context of the cognitive impairment observed in these particular rats. Also looked at plaque deposition to see whether the impaired cognition might be related to plaque formation. So we showed that there was an absence of plaque deposition in the hippocampus and cortex, and this was expected due to the time that it takes to develop plaques. And really what we're seeing here reflects previous findings in post-mortem human brains that cognition and neurogenesis is already altered before extracellular amyloid deposition. Okay, so we wanted then to relate the changes that we saw in the animals, in the neurogenesis, in the cognitive function, back to the human situation. And we were wondering also what might be mediating the effects between the periphery and the brain. So we know, however, there's kind of three pieces of information here that kind of informs what we did next. There are no neuroimaging tools which have sufficient resolution to quantify neurogenesis in living humans. We also know that the neurogenic niche, or the part of the hippocampus where neurogenesis occurs, is highly vascularized. And thirdly, that human blood metabolite signature is significantly influenced by gut microbiota. So to assess the potential of the blood environment in AD patients to modulate or to potentially affect neurogenesis, Our collaborators in King's College in London then used an in vitro neurogenesis assay that they have established and routinely use in their lab. So then the question here was, how does the Alzheimer's systemic environment modulate or affect hippocampal neurogenesis? So they have an embryonic human hippocampal progenitor cell line, and they exposed these cells to serum from control subjects on the Alzheimer's patients. And they measured then or determined the percentages of cells that have markers for neural stem cell proliferation and differentiation as indices of neurogenesis. And so what they saw was that serum from the Alzheimer's patients overall, they decreased neurogenesis, this proxy measure of neurogenesis in vitro. So specifically saw a decrease in Ki67, which is a proliferative marker. In the first phase of this assay under proliferative conditions, when cells then were induced to differentiate in the presence of the serum from the Alzheimer's patients, there was a decrease in MAP2 and DCX, and both of these are indices of differentiating neurons, and that they were this was impaired essentially due to stimulation with the serum from Alzheimer's patients. So we could again then link these observations to a clinical phenotype of Alzheimer's patients. So a positive association between this MMSE or cognition score in our participants with the numbers of DCX and MAP2 or neurogenic cells. So the higher the cognitive ability, the higher the number of these neurogenic cells. In terms then of the link between the gut microbiota in the humans and these proxy neurogenesis measures, the abundance of this pathobuoyant or potentially harmful genera desulfovibrio was negatively correlated with the expression of the neurogenesis marker. So these associations then suggest, from what we can determine, at least in human situations, that impaired neurogenesis may be the link between the altered gut microbiota composition and cognitive impairments that we observe in Alzheimer's disease. So finally then, we investigated what may be mediating the effects between the periphery and the brain, and specifically what may mediate the behavioral and neurogenesis changes that occurred as a result of FMT from the Alzheimer's patients. So to do this then, we examined microbial-derived metabolites. And this field of investigation is getting very busy in trying to determine the links then between the gut microbiota and the brain, and this whole area of the microbiota gut-brain axis and the mediators that are involved. So we carried out untargeted metabolomics, and we found that a number of metabolites were differentially abundant, first of all in the cecal or in the gut content, and we also looked at it in the hippocampus. So firstly, here in the gut content, we saw that there were levels, changes in the levels of histidine, kynureninic acid, xanthrenic acid, and hydroxybutyrate. And the changes we observed reflect what has been reported in Alzheimer's patients and also in AD mouse models of Alzheimer's. And so just to notably then, this metabolite hydroxybutyrate, there was a decrease, just pointing it out here, in the abundance or the levels of hydroxybutyrate. So this is a metabolite of butyrate itself, which is a short chain fatty acid, and it's involved in the metabolism of A beta and tau, the hallmarks of Alzheimer's. It's also been shown to promote neurogenesis in studies in vitro and cognitive function. So it may be a potential link here of what we're observing in our studies. We looked then to the hippocampus, and much fewer differentially regulated or abundant metabolites, only 3 were identified. But interestingly, all 3 of these metabolites, 4-guanidine butyric acid, homocitrulline, and taurine have been shown to be altered in early Alzheimer's patients, especially, and in the mouse models of Alzheimer's. So in particular, we were interested in taurine because it's been shown to be decreased it's the triple transgenic mouse model of Alzheimer's, but also it's been shown to increase neurogenesis. So maybe some of the effects of FMT on neurogenesis and associated behaviors that we see here could potentially be mediated by taurine. Interestingly, taurine is abundant in seafood, and as you know, the seafood a component of the Mediterranean diet and recommended by the World Health Organization to potentially reduce the risk of cognitive decline. So overall, we think that the results here support the idea for a role of these gut microbial metabolites in some of the changes that we're seeing in Alzheimer's. So just to summarize the results then from what I've shown you here so far, the data that we've generated show then transfer of human Alzheimer's gut microbiota to rats caused an impairment in spatial and recognition memory, also caused an impairment in pattern separation type of cognition. And this was associated with a reduction in hippocampal neurogenesis. Also with a reduction in the complexity or the neurite complexity of these new neurons after the Alzheimer's FMT, and suggesting then that the underlying mechanism for a causal role of gut microbiota in cognitive changes associated with Alzheimer's might involve this process of neurogenesis. Changes were also associated with changes in the expression of microbial metabolites. And finally, then, we think that the results from our human serum studies, combined with the results from our FMT or back translation of FMT studies in rats, suggest that neurogenesis may be a central cellular process that regulates both the systemic, circulatory, and the gut-mediated factors in Alzheimer's disease. So in terms then of potential positive modifiable lifestyle factors that can impact upon the gut microbiota, there's evidence now that exercise can increase taxa, the phyla and genera that produce health-promoting metabolites, such as these short-chain fatty acids, including butyrate. So we think that perhaps exercise might act through the microbiota and the gut-brain axis to impact upon hippocampal neurogenesis and cognitive function. And lots of studies now have shown that exercise itself can enhance neurogenesis, and this is in rodent models, and particularly in the aged hippocampus. So seminal work really by Henrietta van Praag has shown this. It's also been shown that various hippocampal dependent behaviors, including this behavior of pattern separation, is enhanced with exercise as assessed using location discrimination tasks. Reports in human studies have also shown the beneficial of exercise on hippocampal volume as a proxy measure for hippocampal neurogenesis, because we can't really determine it per se in the living human brain. And also enhanced pattern separation as assessed by mnemonic discrimination tasks in humans. So we're interested then in the interaction between the gut microbiome and exercise that might bring about changes in neurogenesis and cognitive function. And I'll give you a flavor of the work that we have ongoing in this regard in relation to brain aging and in relation to Alzheimer's. So firstly, Sarah Nicola, she's a postdoc in the group, and she's been working on the interactions and investigating the interplay between the gut microbiota and exercise on hippocampal function. So Sarah has carried out a study where she again depleted the gut microbiota in rats using an antibiotic cocktail. And so that with this microbial depletion, that over a number of weeks, that rats had an impaired performance in this cognitive task of pattern separation. But when rats had been allowed voluntary access to running wheels, this impairment was somewhat attenuated. So she also saw that this antibiotic-induced decrease in the behavior was mirrored by an antibiotic-induced decrease in the neurogenesis, and again attenuated by exercise. So suggesting that there is an interaction ongoing between changes in the gut microbiota, neurogenesis, and behavior. Now, this is in adult rats, and so we're continuing then with studies to look at the effects in a mouse model of Alzheimer's disease. So this is a study which is led by a PhD candidate, Zoe Williams, from Canada, and she's examining the role of the gut microbiome in potential exercise-induced changes in neurogenesis and behavior, and using the 5xFAD mouse model of Alzheimer's. So the hypothesis then that the gut microbiota might be involved in the changes that we're seeing in neurogenesis and in behaviors. So again, using FMT studies from mouse to mouse and also from human to mouse to examine these changes. So as well as Alzheimer's, we're interested in aging in general, and particularly we're interested in middle age. So this figure is from a systematic review and meta-analysis from a publication a few years ago, and it shows and it maps out modifiable factors for Alzheimer's throughout the life course. But what's interesting about how this is depicted is that it shows that middle age is a potential turning point in terms of the relative risk for developing Alzheimer's. So we really see a push-pull, if you like, between the negative and positive factors that impact upon brain health. So we think perhaps that middle age could be a transitioning time or a crossroads or turning point of the lifespan. It's also been shown to be associated with weight gain, a decline in metabolism, in some cases a decline in memory performance or brain processing speed, and perhaps depending on lifestyle influences, then this may precede dementia or prodromal dementia at least. So we have studies ongoing then in UCC where we're investigating mechanisms that may underlie the effects of exercise during middle age. And those studies those factors are related to inflammation, to the gut microbiota. And we're hoping that the results may inform or be predictive of positive or perhaps negative brain aging. So we're currently carrying out FMT studies from middle-aged exercising humans. So this is in collaboration with our partners in London and King's College. They have collected samples now before and after an exercise intervention, carried out cognitive tasks. And these participants are middle-aged, and the samples that we will do the FMT studies into the rats have been stratified based on an exercise-induced enhancement of mnemonic discrimination. And the aim here really is to investigate the causality of exercise-induced changes in the gut microbiome on again, looking at the neurogenesis and behaviors in our rats. And so we're hoping that by researching this role of the microbiota gut-brain axis in shaping these memory changes during middle age, we might be able to develop information about a potential gut microbiome signature that could be predictive of memory decline potentially in later life, or not, depending how the results come out. So really, to finish, I'm going to leave you with this figure, and it's from a commentary in Science last year by my colleague John Cryan here in Cork, together with Sarkis Masmanian. And in this commentary, they've outlined their thoughts on advancing human microbiota research, which would then hopefully inform the causality of the gut microbiota in brain and behavioral changes. So amongst the various recommendations is performing large-scale translational and longitudinal studies And really in the context of Alzheimer's and brain aging, this would be very informative. Another area is to understand the effect of lifestyle, and including that of diet and exercise. And this also then links back to our modifiable risks identified for dementia and Alzheimer's prevention or delay. So finally, I would like to acknowledge my research team, and in particular, Dr. Stephanie Gabruker, who led the work that I've described on the Alzheimer's FMT, Dr. Sarah Nicola, who is a super postdoc doctoral researcher in the group, and the past and current lab group members, the technical support, and to acknowledge the funding in particular by Science Foundation Ireland for most of the work that I've presented here today. Thank you.
Thank you for a very interesting talk. Well, I'm waiting for some questions to come into the Q&A box. I just have one question regarding the taurine work. Because as you mentioned, fish is a reasonable dietary source, but in addition to diet, I mean, we have an endogenous production, and this, in what role might the gut microbiota actually play in taurine production, if any?
So we saw it in the hippocampus. The metabolite was affected there. I guess, you know, there are so many different classes of microbes in the gut that the microbiologists are still trying to figure out which are the good and which are the bad, if you like, and what are the influences by so many factors that can affect these. So how do the gut microbiota produce the taurine? You know, it is a metabolite and can be produced within the brain, within the gut. But the mechanism really isn't worked out, not to my knowledge at least anyway. We might have to defer to the microbiologists for that one.
I know this as yet there are no questions that I see in the Q&A. I guess we can wait for another little while to see if any occur. Just in terms of well, waiting in terms of your future work, you've mentioned some of the work you're doing in middle-aged individuals. What else? What other types of studies you're planning right now to further investigate the role of the microbiota?
In the middle age. So I guess the middle age studies that we're doing, we have a series of studies where it gets independent of the gut microbiota and then including the gut microbiota. So what we're focusing on looking at proteomic profiles in the blood and in the brain and the CSF from our middle-aged, we have middle-aged rats to do this. Because obviously in the human situation, we kind of take out the human brain to have a look at. So what we want to do is to create, to look at what converging factors between the blood, between the CSF and between the brain in middle age in our rat models. And if we can determine something that could be picked up in blood, so to speak, that it could be potentially then translated to the human situation in middle age. Then if we come in with an exercise intervention, can we find a protein or metabolite, is a question that we're interested in, that could be modifiable by exercise? And so could be potential signature or marker that could be measured during middle age, and then maybe, you know, could be a predictive of how we age. And linking each of these signatures then with cognitive function and with neuron function is really how we would underpin what's happening. If you put the gut microbiota into the mix there's another peripheral level of information that can be generated. And I think in future, you know, there's a lot of interest now about how can we determine a gut microbial signature, so to speak, that could, you know, determine how healthy we are, because of the influence not just of you know, our environment and our diet and exercise, further environmental influences. Stress has a huge impact on the gut microbiota composition, as does air toxins, as does pollution. So, it is quite complex. Geography also has an impact on our gut microbiota composition. And interestingly, some work now is going on looking at social interactions and how that can impact. But I suppose it's all intertwined, because if you're socially connected, you may be potentially less stressed, or you may be more physically active, which then will complement the potential benefits on the gut microbiota.
Okay, thank you. During that time, the Q&A box has populated with a number of questions. So let me ask the first question here from Thomas Chin. And he thanks you for a very interesting topic. And notices that in an earlier slide, the control humans had a BMI of 25, while the AD had much higher. Was there any study of the BMI effect on the microbiome?
We didn't do that, but there is evidence to suggest that obesity has a negative effect on microbiota composition. So that might have determined that. I think perhaps when we think of Alzheimer's patients and of their potential living conditions and maybe potentially sedentary behavior, that might contribute potentially to an increased BMI. So that might be having a role there as well.
Yen who asks, what are the cohorts that include early life modifiable risk factors for Alzheimer's disease? Then questions, are most studies on Alzheimer's started from midlife?
So, sorry, you repeat the start of that one again?
The second part is just is parenthetically. The first one is about cohorts that include early life modifiable risk factors for Alzheimer's. And then parenthetically, she asks, but she states, most studies on AD started from midlife with a question mark. I guess the question is really where do most of these studies really start when you're looking at these cohorts of Alzheimer's disease?
Yeah, so I think with regard to the early life modifiable risk factors, well, education has really been the one that's been highlighted, and education in early life as a potential, it's a very strong modifiable risk factors for dementia. I think what's missing from a lot of the Alzheimer's field, especially in the context of the gut microbiota, are these longitudinal studies. So the information really isn't there to determine how the gut microbiota influences over long periods of time. I mean, the study we did here, we had clinically diagnosed Alzheimer's patients who were in an older aged cohort of life. There are studies ongoing now on MCI, or mild cognitive impairment, which precedes Alzheimer's disease. And so there's more evidence coming out that in MCI, that there is perturbances, if you like, in the gut microbiota that could potentially influence cognitive behavior, etc. And again, going back to our work on middle age, I think that's really, there's no cure for Alzheimer's is the problem and is the issue. And I think by focusing on earlier times of the lifespan, like middle age, and by the influences of environment and lifestyle here, to delay or reduce the risk as much as we can of developing dementia is where I think research would be very importantly positioned.
Jennifer Lee asks, first, thanks you for a wonderful talk, and says you showed data that the number of goblet cells is reduced in mice receiving FMT from Alzheimer's disease patients. Did you measure mucus thickness or gut barrier function? Is there a difference in gut permeability?
We didn't measure that, but it's a very good point and a very good question. So yeah, I guess we're the goblet cells are the proxy measure of the gut barrier function protection, but we didn't look at the permeability per se. in our studies, but it definitely would be something to consider for future studies.
The next study, I think, next question is very similar, and I think you've answered it, but I'll ask you just to make sure. So, the Thomas Chin asks, any study on mucus levels in the gut protecting the gut cells?
Yeah. So, so we didn't do it in this study. Let me think. Can I think of? So this is the first time we've done an Alzheimer's FMT into an animal model. Most of the FMT studies in human that are used at least for therapeutic purposes are in the area of C. difficile, ulcerative colitis, and there have been studies there done where they're looking at the mucus levels and the gut function. So no, so to answer your question, it's the same answer as the previous one. We didn't measure the mucus in our animals, just the goblet cells.
The next question comes from Larissa Constantinou, and she asks, could antibiotics have negative effects on adult hippocampal neurogenesis?
Yes, they can, and they have. So the antibiotics study that I showed you that Sarah did, we saw that there's an impairment in neurogenesis. Now, this was a cocktail of antibiotics that was given over a period of a number of weeks consistently. So the idea here was that we were to deplete or create a dysbiosis in the gut microbiota, and it did have a negative effect on the neurogenesis. I think when we consider antibiotics, it's really the dose and the type is really, really important. For our FMT studies, we used an antibiotic cocktail, a very different one, to deplete the host microbiota of the rats in the first instance, in order that we could engraft in then the microbiota from the Alzheimer's. But that was a kind of a one shot at the start, and to allow the engraftment then from the FMT. Whereas Sarah's study, and there wasn't an effect of the antibiotics on the neurogenesis in that study, because we have done kind of trial studies, if you like. So that short Alzheimer's FMT, which was a short antibiotic application, where at the start of the study we measured neurogenesis later, it didn't have an effect. However, if you have a long, prolonged administration of antibiotics, it does negatively affect neurogenesis.
Next question comes from Ed Salzman, and he asks, how important are the dietary substrates, example, fermentable fiber or carbohydrate, on the putative link between gut microbes and brain function?
Yeah, so there's a lot of work in this field looking at fiber, looking at prebiotics, probiotics on brain function, both in animals and in humans. So the evidence to date suggests that probiotics are beneficial bacteria, so your lactobacillus, for example, and there has been evidence to show that these probiotics can have beneficial effects on cognitive function. What's interesting now is some of the work going on in the context of prebiotics. So these would be elements that can help develop and promote beneficial bacteria. So things like oligosaccharides, which help increase the abundance of these beneficial bacteria. Inulin is another one. And more recently, work has been done now on the polyunsaturated fatty acids, or PUFAs. So are they proposed as a type of prebiotic to help increase the abundance of positive bacteria and microbes in the gut. So yes, there is evidence to suggest that they have a beneficial effect on cognitive function and on brain function.
Okay, and Huan Wang asks, in older patients with dementia and diabetes, can you tell if disease status affects the composition of the gut microbiota?
So I guess it's a bit of a chicken and egg, which comes first. I don't know if there is a differential effect of the diabetes combined with the Alzheimer's that would make it worse, if that's what the question is about. We do know that with diabetes, there is a dysbiosis of the gut microbiota. We know that it occurs in Alzheimer's. We also know that they both can often occur together. And some of the patients in our cohort did have diabetes as well. But to separate out of all of those, it does require, I suppose, controlled studies and longitudinal studies as well.
And Cal Lai asks, is there any experiment done on fecal microbiota that transfer from healthy humans to mice to reverse Alzheimer's?
So there have been mouse-to-mouse studies, but there hasn't been any study from a healthy human into an Alzheimer's mouse model. No, so there's been one study done by my colleagues in Cork where they transplanted fecal microbial matter from healthy mice into aged mice. And the age-related deterioration in cognitive function and memory was reversed with the transfer from the microbiota from the young mice. So young into age increased or reversed the cognitive impairments. But there hasn't been anything done from a healthy human into an Alzheimer's mouse. But that might be our next study.
And the final question, is there a typical gut microbiota profile in Alzheimer's patients compared to healthy controls?
So from just the earlier slides that I showed, there is some evidence to suggest that the balance between two particular phyla of bacteria, the Bacteroides and the Firmicutes, is impaired or is dysregulated in Alzheimer's. So yes, just recently in the Journal of Neurology, there was a systematic review, and they looked at, I think it was 34 cases of Alzheimer's patients, and so that there was a consistent change in these two types of bacteria. So it might feed into what we're thinking of as a potential profile. Now, these are the most abundant phyla in the microbiota, so they could be affected in other conditions as well. But I think we need to look a little bit more closely at the genera, which are the kind of the next level of taxa, to see if we can really pin down a specific signature. And then I suppose next step is, can we therapeutically target this in the future?
Okay. Thank you very much for a wonderful presentation and for addressing all those questions.
Thank you, Paul. Thank you very much, and thank you for your attention and for listening.